Dear audience, Assalamu alaikum. I am Professor Shamsud-Jaman, Professor of Pathology. I welcome all in today's 17th lecture on hematology. Today's topic is hazards of blood transfusion and incompatibility of blood transfusion. Hazards or complications of blood transfusion and incompatibility of blood transfusion. Before going to discuss about this, we have to recapitulate blood grouping. Now come to blood group. Dear audience, we know we have ABO blood group and RH blood group. RH rhesus blood group. Respected audience, we know blood group is based on antigen present on red cell membrane. Based on antigen present in red cell membrane. The antigen appears early in the fetal life and it remains unchanged. You know, antigen appears early in fetal life and remain unchanged. Nature of antigen. Nature of antigen, it is glycoprotein. Or glycolipid. In nature. Now come to ABO blood group. ABO blood group system. Blood group A. Blood group A. In case of blood group A, there is antigen A on red cell membrane. Antigen is A, and in the serum the antibody is anti-B. Anti-B antibody. So the individual who is with blood group A, his antigen is A on red cell membrane and anti-B antibody in serum. Blood group B, the antigen is B and antibody in serum is anti-A. anti-A. Blood group O, here the antigen on red cell membrane is O antigen. Antigen is O and antibody in serum is anti-A and anti-B antibody. Blood group AB, antigen is AB and in the serum there is no antibody, no antibody. So this is ABO blood group system. Now come to RH blood group or research blood group. Antigens are C antigen, D antigen and antigens. So capital C and small c, capital D antigen, capital E, small e antigens. Among these antigens, D is strong. D is strong antigen. So blood group RH positive means RBC membrane contains antigen D. RH negative blood group means there is no antigen D in RH membrane. RH positive means presence of antigen D in red cell membrane. RH negative means no antigen D in red cell membrane. Now come to antibodies. The antibodies may be naturally occurring antibody and acquired antibodies. These may be naturally occurring antibodies. Like anti-A, anti-B, and acquired antibodies. The acquired antibodies are formed due to presence of antigens. Example, anti-D antibody, When Rh negative individual is transfused with Rh positive blood, there is production of acquired antibodies, that is production of anti-D antibody. How produced? How produced? When Rh negative individual is transfused with Rh positive blood, then there is production of acquired antibody like anti-D antibody. How? You know, Rh negative means no antigen, no antigen D. If this individual is transfused with Rh positive, is transfused here, what will happen? The Rh negative person gets antigen D, gets antigen D. Gets antigen D, and due to this presence of antigen D following transfusion in Rh negative individual, there is production of antibody against this D, and this is anti-D antibody, and this is acquired. Another way of formation, entry of Rh positive red cell, entry of Rh positive red cell from fetus to Rh negative mother during delivery. If during delivery Rh positive red cell from the fetus enters into Rh negative mother, then in Rh negative mother there will be production of antibody against D, antigen D. It is nothing but acquired antibody, that is anti-D antibody in Rh negative mother. Now come to hazards of blood transfusion, that is complications of blood transfusion. The hazards may be febrile reactions, allergic reactions, there may be hemolytic reactions also. Then transmission of disease. Dear audience, you know, following blood transfusion, certain diseases may be transmitted from donors to recipient. What are those? Like hepatitis B virus, hepatitis C virus. circulatory overload. Following excessive blood transfusion, there may be circulatory overload also. Thrombophlebitis, there may be air embolism also. Dear audience, you know at least 100 cc of air or more than 100 cc of air is needed for air embolism. So, these are the hazards of blood transfusion. Now, come to incompatibility of blood transfusion. Incompatibility of transfusion. It is of two types. One is ABO incompatibility and another is RH incompatibility. First come to ABO incompatibility. Dear audience, if there is mismatched blood transfusion, there will be ABO incompatibility, and in case of ABO incompatibility, the donor's blood and also the recipient's blood undergo destruction. Again, following ABO incompatibility or if there is mismatched blood transfusion, donor's red cell undergo destruction and also recipient's red cell undergo destruction. So, destruction of red cells in donor's blood and also in recipient's blood. Now come to red cell destruction in donor's blood in case of ABO incompatibility. Suppose the donor is with blood group A. Here antigen is A and antibody is anti-B. Recipient blood group B. So, here antigen is B and antibody is anti-A. Dear audience, if the donor is with blood group A, he has antigen A on red cell membrane and he has antibody anti-B. If the recipient is blood group B, antigen is B on red cell membrane, antibody is anti-A. If such blood group A is transfused to recipient having blood group B, there will be mismatched blood transfusion and there will be ABO incompatibility, and there will be destruction of donor's red cell. How the donor's red cell is destroyed here? How donor's red cell undergo destruction? Suppose this is the donor's red cell containing antigen A, and this is the recipient's serum containing anti-A antibody. This antibody of the recipient will binds with this antigen. So, complex is made by recipient's anti-A antibody with antigen A present on donor's red cell. Again, the donor has antigen A on red cell membrane and the recipient's serum has anti-A antibody. A complex is made by recipient's anti-A antibody with antigen A present on donor's red cell, and this complex activates complement system, by classical pathway. This antigen antibody complex, that is immune complex, activates the complement system by classical pathway, and there will be formation of membrane attack complex, and this membrane attack complex causes lysis of red cell membrane and hemolysis of donor's red cell. So, this is the mechanism of destruction of donor's red cell when there is mismatched blood transfusion. Now come to what happens in recipient's blood. Dear audience, you know here the recipient is blood group B. Blood group B. So, here antigen is B and antibody is anti-A. If this is the red cell of recipients, there is B antigen, and if the recipient is transfused with 1 bag of blood, 1 pint of blood or 350 ml of blood, the 350 ml blood of donor blood contains anti-B. This anti-B should react here and should hemolyze, but fortunately this antibody is diluted in the recipient's blood. So, usually there is no hemolysis in recipient's blood. So, recipient's RBC destruction is less important here. Dear audience, now come to RS incompatibility. Dear audience, I have told you RS positive blood has antigen D and there is no antibody. If RS positive blood into RS negative individuals, there will be production of anti-D against the antigen D. Suppose mother RS negative. It means mother has no antigen D. And this is fetus. Suppose fetus is RS positive. What it means? The fetal RBC contains antigen D. During first pregnancy, what will happen? The fetal blood containing antigen D will enter into the mother's circulation, and as it enters in mother's circulation, due to presence of antigen in mother circulation, there will be production of anti-D antibody. Again, if mother is RS negative, fetus is RS positive, during first pregnancy, fetal blood containing antigen D on red cell membrane will enter into mother's circulation, and within the mother's circulation there will be production of anti-D antibody against this antigen coming from fetus to mother. Initially, this antibody is IgM, and due to high molecular weight, IgM cannot cross the placenta to enter into the fetus. Again, initially there is production of IgM antibody, and due to its high molecular weight, it cannot cross the placental barrier to enter into the fetus from mother. In subsequent pregnancy, if the fetus is again RS positive, there will be destruction of red cell in fetus. There will be destruction of red cells in fetus. How? In subsequent pregnancy, mother RS negative, but mother has got anti-D antibody, and it is IgG in nature. So in subsequent pregnancy, mother is again RS negative, but mother has anti-D antibody which is IgG in nature. If this is the fetus, RS positive and has antigen D in RS, antigen D in red cell membrane. From mother, the antibody IgG will cross the placenta and enters into the fetal blood. And this IgG coming in the fetal circulation binds with this antigen present on the red cell membrane. So, mother's IgG anti-D antibody binds with D antigen present on fetal red cell, and formation of immune complex, activation of complement system, by classical pathway, and formation of membrane attack complex, and this membrane attack complex causes lysis of the fetal red cells, and there is destruction or hemolysis within the fetus, and it is called erythroblastosis fetalis. It is called erythroblastosis fetalis. It is called erythroblastosis fetalis. Dear audience, again, if mother is Rh negative and the baby is Rh positive, in first pregnancy, the fetal blood containing antigen D on RBC membrane enters into the mother circulation and stimulates for production of anti-D antibody. Initially, the antibody is IgM. Due to higher molecular weight, it cannot cross the placenta to enter into the fetus. In the subsequent pregnancy, if the fetus is again Rh positive, in the cessation from mother's blood, IgG antibody cross the placental barrier and enters into the fetus, and this anti-D antibody binds with the fetal antigen D present on the RBC, and there is formation of immune complex, activation of complement system, formation of MAC membrane attack complex, lysis of fetal red cell, and this is nothing but erythroblastosis fetalis. Now come to prevention of such type of Rh incompatibility. Prevention of such type of Rh incompatibility. Administration of anti-D immunoglobulin. Administration of anti-D immunoglobulin in mother in first pregnancy. Within 72 hours of delivery. If we can administer anti-D immunoglobulin in mother in first pregnancy within 72 hours of delivery, this anti-D immunoglobulin will prevent the immune response to produce to produce anti-D antibody in mother's blood. So this is the prevention of RS incompatibility, and this is all about the RS incompatibility, ABO incompatibility, hazards of blood transfusion. Dear audience, today up to this, thanks all.